all agree that the cell on your page, if it were in interphase, would look like this. It wouldn't be X's because DNA did not duplicate itself. And this cell in interphase is a cell that just happens to be going about its life processes, right? Like doing whatever it is intended to do as its adult form. That means this contains all of the DNA that it needs, right? What do we call it when it contains two copies of everything? One that I got from mom, one that I got from dad? So it does have homologous chromosomes. They're lined up next to each other. If homologous chromosomes are present, we use a mathematical representation of it. What is it? 2n. What does it mean to be 2n? This is a diploid cell. That means it has two copies of all of its chromosomes. Just so happens that this poor cell only has one chromosome. Has two copies of all of its chromosomes. That means it has two copies of every gene. Yesterday you saw the genes at the bottom. And those genes were labeled and they were in the same spot on both of the homologous chromosomes, right?
Wait, is one of these from each parent?
Mm-hmm.
So
this would be like any adult body cell. So say if I took one of your skin cells, it'd be just like that. So knowing that it's just like that, it means it has two copies of everything. And, well, these should be the same size, but my fine art skills, they did not end up the same size. That means that they both contain the exact same genes. What's a gene?
section of DNA that goes to protein.
Mm-hmm. But just because they're the same gene doesn't mean it has to be the same recipe, right? What do we call different recipes for the same gene? It's an allele. So an allele is versions, different versions of the same gene. So one parent could have given capital A as the version of the gene here, and the other parent may have provided lowercase a. But at the same time, one parent could have provided capital A and the other parent could have also provided capital A. Or both parents could have provided lowercase a. It depends on what they had to give. You can only give what you have, right? There's no way to give anything that you don't have. So they had two choices of what they could give you. And because of meiosis that you saw yesterday, a cell like this separated, made gametes, and then what you get, well, it's kind of luck of the draw. How many of you agree it's just a coin toss what you get? That's how genetics works. It's a coin toss. If there are two different copies, heads or tails, 50-50, you don't know what you'll get. Even if that means your parents have two lowercase a's, one from their mom and one from their dad, it's a coin toss whether you're going to get the one from your grandfather or your grandmother. Does that make sense? Now we'll take a look at an actual human chromosome labeled. This is the X chromosome. And I want you to look at this X chromosome because I want you to see a couple of things, one of which being this indent right here is where the centromere would attach. So if this duplicated, you would get a centromere right there. So when geneticists talk about a chromosome like this, we call this the short arm because it's the short one, and this the long arm of the chromosome. So we have a short arm and a long arm. The other reason that we look at this is because it's all stripy. In real life, your chromosomes are not stripy. This is a map, it's called a cytogenic map, that geneticists use to locate genes on specific chromosomes. And unfortunately, all of the genes that are labeled on this chromosome are actually disease-causing genes. Congratulations, your X chromosome, these are all of the diseases you could have inherited. How you feeling about that coin toss now?
Pretty good. Pretty good.
Here's the worst part of that coin toss if you are not a girl. If you're a girl, it's still 50-50, life is good. If you are one of the gentlemen in the room, you only got one of these. So, well, while it was a coin toss, which one you got from mom, because you got your X from your mom, whether or not you got the healthy X or a sick X, well, you only get one shot at this thing. Ladies, you got two, because you have two X chromosomes. So what that means, let's take a look at some of these particular diseases here. How about, how many of you have heard of Duchenne muscular dystrophy? Muscular dystrophy, you've probably heard it, seen it, at least seen advertisements for the Muscular Dystrophy Association. It's a genetic disorder on the X chromosome. Ladies, you need 2 copies of the disease allele. So there's a healthy allele. We all have that gene. Every single one of us possesses every single one of those genes. It's whether or not you get the good copy or the bad copy. The one that makes you healthy or the one that makes you sick. That's one of those diseases that requires 2 of the bad copy. If you're a girl. If you're a boy, you only get 1 copy. So if you got the bad copy, sorry. Make sense? Some other pretty rough diseases on here. Turns out, first hour did not care for me pointing out, which one was it over here? Oh, Lesch-Nyhan syndrome. Inability to digest one of the amino acids causes you to pee out solid crystals every time you pee. They didn't like that one. They were all like, oh. You all look disturbingly uncomfortable. It's like passing a kidney stone every time you pee. How are you male and female? Yeah. So there are, in a human, we all agree there are 23 different chromosomes, right? Everybody agree? Of those 23, the first 22 have absolutely nothing to do with whether or not you're a boy or a girl. Those are autosomes, we have them for all the other traits. The X chromosome, there are the last set of chromosomes, I should say. The 23rd pair, there are 2 choices of the kinds of chromosomes you may get. You can get an X, which is a nice full-length chromosome, has all these lovely genes on it. Or you can get a Y. Y, sorry guys. See how lovely and large this X chromosome is? The Y chromosome is about that long in comparison. And it really only contains one relevant section of genes called the sex-determining region on Y. And that doesn't make anything extra. All it does is turns off genes that are located on the X chromosome. So the only reason males are male is because some of their genes got turned off by that truly dinky Y chromosome. True. And the Y chromosome just keeps getting smaller and smaller and smaller with every passing 1,000 years. So, give it another few thousand years and it's going to be even smaller. There are some scientists who argue that it's going to disappear altogether. Not to give you guys a complex. The only one that's going to be left is going to be X, but it's just us.
So if guys only need one of those genes, to have that disease, are they 2 times more likely to have it than girls?
Yes, all of these diseases are considered significantly more likely in a male. Because all of these diseases, if you only get 1 copy, There are some of them that are dominant, like on here, I do believe Huntington's disease is on here somewhere. It is a dominant disease. That means that all you need is one copy of it to have the disease. So it equally affects males and females because females only need one copy.
So if you female, you get like one bad, one really bad, and you get another one.
You're healthy.
So as long as like you get two bad ones.
Yep. Only problem would be is if you're female and you get one good one, one bad one, is that you have a 50% chance of producing an offspring who has that. So you gotta consider that.
So because we have a bunch of genes on here, bad ones,
Huh?
does that mean we're like, there's like a 40 and 1 chance that we're gonna get one of them?
No, because of the frequency with which the disease gene occurs. A lot of these genes that the bad versions are not very common in the population. Some of them, like Lesch-Nyhan syndrome, is very uncommon because besides that whole pea and crystal things, it kills you when you're very young. So you die before you're going to reproduce. If you die before you're going to reproduce, the allele frequency, the how common that is in the population, decreases because you can't pass it on. That's kind of, a bummer. Other fun ones on, yep. Yes. Yep. Females can only give X chromosomes because all we have are X chromosomes to give, right? If we were doing that whole meiosis thing with the X chromosomes, female, it's an X and an X, and when it is separated to opposite sides of the gamete, we end up with X or X. Gentlemen are X and Y, which is why, if you've ever heard someone complain about the gender of the child that they're going to have, typically it's always a father who is bummed out that he's not having a son. It was his fault. So he was the one who had that choice. My sister is expecting in September. It is her 4th child, and it is her first little boy. So my brother-in-law is finally super excited. We were like, good job, guy. It was all on you the first time. So now I just want to know what they're going to name it, because the first 3 were A's, Amelia, Alice, and Abigail. Do they switch to the next letter because it's a boy? Or do they stick with A's? Because I started listing A's. I was like, Andrew, Alphonsus, Abernathy, and they were like, stop naming children. And I was like, those are great names. And you're like, if we want our child, beaten on the playground, those are great names. And I was like, Andrew's a solid name. You might want to avoid Abernathy. No, my sister's one of those people who gave all of her kids 2 middle names. I have no idea.
Makes the paperwork harder.
I don't know.
Were they all same middle names?
No, they're all completely random. I never purported that she was the most logical individual. It's just they're all A's. But she's like, well, when they were all girls, it just made it easy, because then all the embroidered stuff with A's on it, I could just pass down. I don't know. I'm just hoping I didn't get that half the gene pool. Because we're siblings. That means half our genes are in common. There are some other interesting things on here. Albinism, so whether or not you make skin pigment located on the X, yes, that means it's more common for boys to lack skin pigment. Cleft palate, I think, is on here. You guys know what cleft palate is? When you develop in utero, you develop in perfect symmetry. Do you ever notice that if you just drew a line down your body, that your body would be like a mirror image of the other half, right? So when you're developing, you develop in perfect symmetry, which means things start on this side and then they start to fuse into the middle. When you come out, you're like all squishy, right? Like your bones haven't developed, you're mostly cartilage. For those of you who aren't really sure why, think about the size hole you have to get out in the size of you. You've got to be kind of collapsible. So your bones don't fully form so you can collapse, especially your head. Your head's got to squish in there to get you out. How many of you feeling really good about that now? They're like, yeah, you're squishy. Be careful with the heads of infants. You're squishy. And one of the last things to form in that whole process of you being squishy and symmetrical is the roof of your mouth. That's called the palate, the roof of your mouth. And if you have cleft palate, you have a gene that's on the X chromosome that prevents it from fusing shut. So it's empty right up through the middle of the roof of your mouth. And it's empty, there's no skull right here. Your skin doesn't form around skull because there's no skull to form around, so you have a hole right there. This is really bad because think about how infants feed. Infants feed by requiring suction on a bottle, right? If you have a hole there, you can't eat. So babies, when they were born with cleft palate way back in the day, it was pretty much a death sentence because there was nothing they could do for them. Now we have all of these really amazing surgeries where they can fuse that shut, but it's like years of surgery through their childhood as their head begins to develop and grow to make sure it gets fused shut properly. It's on the X chromosome. More common for a boy to have cleft palate than a girl because of that, because a girl, 2 shots at this thing. Guys, you only get 1. That's why it's really important that we can draw these kinds of cytogenic maps so we know which genes cause which traits. Not all traits are diseases, but this one's diseases because sometimes it's interesting to look at how a very tiny change in DNA makes a big change in appearance. It's a very tiny thing, but having a completely fused palate, sort of a big deal. Don't worry, people with cleft palate now, they survive. Minor scars, usually noticeable if you know what you're looking for, but there are some amazing oral and maxillofacial surgeons who can work wonders. Children's Hospital, University Hospital, they're all very good at those things. These are all really sad because they're all diseases. I don't really feel like talking about sick people anymore. Like ocular albinism isn't that big of a deal. You just look like one of those red-eyed rabbits instead of like having people-colored eyes. I mean, all things considered, a red-eyed rabbit's not that. Like, oh, okay, so you also can't look into the sun. But we all probably ought not stare into the sun anyway. Agreed? How many of you are like, I stare into the sun all the time. Don't do that. Don't do that. Real bad for your retinas.
I've done it once.
It hurt, didn't it?
It didn't hurt that. It was just like, it was kind of blue for a while. It went away.
Good. Good. That's good. So, there are some of you who genetics thing told me about this guy. Now, some of you guys don't remember Gregor Mendel because I had some people in first hour and some people amongst you who didn't write him down. In first hour, I was like, yeah, you guys know Gregor Mendel, and I had some students who were like, nope. So I'm going to give you the Cliff's Notes. I didn't start with him on purpose. I started with what you already know about genetics, and the reason that I did that is because you know a lot that Gregor Mendel did not know. However, Gregor Mendel is considered the father of genetics, and being the father of genetics, we should probably give him a little bit of credit here. Those of you who wrote Gregor Mendel down on your paper, what was he? He was a monk, right? Lived in the monastery in Austria. And what a lot of people fail to understand about monastic life, they take a vow of poverty. Vow of poverty means they promise that they're not going to accumulate any worldly wealth that is unnecessary to their existence. And it also means that they are going to be self-supporting as a monastic community. That means that they're going to produce something that can be sold at market, not for profit, but just to cover the necessities of life. And in order to do that, they grow a lot of their own food. They have gardens. They grow fruit, they have orchards, and they kind of assign monks living in community to specific jobs based on their strengths. And Gregor Mendel, one of his strengths was that he was trained in science. So if you have a guy who's trained in science, specifically biology, it makes a whole heck of a lot of sense to probably put him out in the garden to grow your food. One of the foods that you would have grown if you were living in a monastic community in the late 1800s were peas and beans. Things that can be dried out, saved over winter, and used over winter. Makes a lot of sense. But Gregor Mendel was no slouch in the intellect department. And in all of his growing, he recognized that there were a lot of traits about pea plants that there were two choices. For example, pea plants were either tall or short. There weren't any medium. Or they were, the pea itself, not the pod, but the pea was either yellow or green. The pod was either inflated or constricted around the pea itself. The flowers themselves either occurred at the tips, so if I'm a pea plant, this would be the main stalk of the pea, and these would be my branches. The flowers would either occur at the tips of the branches, we call that terminal budding, or they occurred at the armpit, where the branch and the stem meet. That's called axial budding. And it was always either or. They were never both. They were never a weird combination of the two. It was never like a half smooth, half wrinkly pea. It was either they were all wrinkly or they were all smooth. But he also knew something about pea plants. They are self-pollinators. So in other words, they don't need bees, they don't need birds, they don't need any of that. The way that the pea flower is shaped is it bends back on itself and it's able to pollinate itself. That means if you allow that to happen, tall pea plants, when their pods are saved, dried, and then replanted the following season, only ever produce tall offspring. And short pea plants only ever produced short offspring. Yeah, it's an observation. That's interesting. This is how we know he was clearly a man of God. Only a man of God would be this unbelievably patient. Gregor Mendel decided to not allow the pea plants to self-fertilize. Oh no. He took a paintbrush and he fertilized the short ones with the tall and the tall with the short. We call it a cross-pollination. But he had to do that by hand with a paintbrush. Every single flower. How many of you are like, I would have quit? I'd have looked at the rector of the monastery and been like, peace guy, I'm done. Unfortunately, he took a vow, so he wasn't allowed to just piece it and leave, but God bless him. At that point, I'd have been done. Even if you consider every one of those flowers a prayer, good grief, you'd be there all year. So he did that, and something weird happened. You would expect medium plants. Tall plus short. Medium. They were all tall. So he saved the seeds, he dried them out, he planted them, all tall. Okay, if I had gotten that result, I would have quit. Gregor Mendel, clearly, again, man of God, he did not quit. Or maybe he did quit. Because he allowed those to self-fertilize. No more of that paintbrush nonsense. He let them self-fertilize and then grew those peas after saving and drying them. Sounds like an inordinate amount of work. But he discovered in that second filial, hey, Latin student, second filial. In that second filial, he discovered that 75% of them were tall and 25% were short. And it was always that very specific ratio. Now, I don't want you to think that Gregor Mendel was following science fair rules and using 100 plants. I mean thousands. Over and over and over and over and over and over. And over and over and over and over again. Man of God. But even men of God are human. He wrote up his results. He published his results. He shared them with the wider scientific community. Because did I fail to mention what his job outside of the monastery was? Because they had to earn a little bit more extra money, so they sent him out to do something he was skilled to do. He was a biology teacher at University of Vienna. And so he published with the scientific community his findings, and he said, okay, there is a character, he called it a character, that was his word, that is passed from parent to child that controls traits. What would we call characters now? Genes. Here's the problem. He didn't know anything about genetics because it didn't exist. No DNA, no meiosis, no mitosis, no chromosomes. That's all like 1952. And Gregor Mendel's like all 1880. So this is how we know that he is in fact human. He quit. He looked at the world, and after they rejected his ideas, balked at him, said he was a fool, said it wasn't possible, he stopped working in science and became an administrator, a rector of the seminary, or of the monastery. So basically what that means is he became a house mom to a bunch of monks. Because he's like, all right, you don't believe me? Fine. Well, the world made up for it later and named the entire branch of genetics for him, which is why we call it Mendelian genetics. Because, well, turns out he was right. He's the guy who allowed us to write these things. And he gave his characters names, capital letter for the trait that he saw more frequently, and a lowercase letter for the one he didn't see as often. So what he did was said, well, the character of my pea plants that are tall, they have only tall character, and that's all that they will ever give to their offspring. And then he took the short pea plants and said, short is the only character they have to pass on. And his test cross, his cross-pollination, allowed him to explain why all of the offspring were tall. Because every one of the produced offspring contained the tall character, which masked the presence of the short character. Now, these things in these boxes we call genotypes. And a genotype is simply what two alleles an organism possesses. So parent number 1, capital T, that's its genotype. Those are the two alleles it has, two capitals. And we can call that homozygous dominant. Homozygous meaning the same zygote, so the same things. The genotype of all of the offspring, we can do a ratio. He loved his statistics, so he said 4 of 4 would be capital T, lowercase t. That's its genotype ratio. 4 of 4, capital T, lowercase t. But remember, Mendel couldn't see inside of the pea. He had no idea what was going on in the nucleus. He had no idea what chromosomes were doing, how they were assorting at the metaphase plate. Couldn't see the spindle fibers pulling them to opposite sides of the cell. All he could see were the traits that those pea plants possessed. Those traits are called the phenotype. Phenotype is what you see, what you perceive when you look at an organism. Or, in other words, the trait that you observe. That's its phenotype. The phenotype of all of this first filial, if these are the parental generation, the capital T, lowercase t, or lowercase t, then our inside of our boxes we label the first filial, or F with a subscript of a 1.
So what's the definition of genotype?
Genotype are the two alleles an organism possesses, whereas the phenotype is what you see when you look at the organism. The phenotype here is 100% tall. Tall is the phenotype, what you see. The genotype is what the genes say, capital T, lowercase t. These offspring in this box, what would happen if you allowed it to self-fertilize? So cross them. With your partner, cross the second filial. the second filial, cross the first filial results, and do a phenotype and genotype ratio. You can talk about it with your neighbor. What did you write across the top? What are the two potential alleles?
And then lowercase t, and on the side is.
I cannot write with the laser pointer. That does not work. After a few seconds, though, it's...
The side is capital lowercase.
So in my first box, homozygous or heterozygous? Homozygous dominant or homozygous recessive? My second box, either next to or below. Let's do both at the same time. And my last box? So we know my genotype ratio, 104 homozygous dominant, 204, also known as 1/2 heterozygous, and 104 homozygous recessive. That's my genotype ratio. I'm sorry, my phenotype ratio.
75% off.
Congratulations, you just did all of Gregor Mendel's work. Well, okay, not quite all of it. Oh, I don't know. You just did what we call a monohybrid cross. Hybrid means combination of two opposing traits, right? Mono means one. So a monohybrid cross would be if I were to take, here's one, here's the other, here's one, there's the other. I think you're probably prepared to do what Gregor Mendel did. He observed two different traits in the same plant. First, we'll look at height, tall versus short, and I have a heterozygote here. And we're going to cross two heterozygotes. So just like we just did in our second filial for our second filial generation. But he's like, yeah, that's interesting enough, but what happens if I took a heterozygote at the loci for tall and crossed it with a heterozygote for yellow? So now I'm looking at two traits, a dihybrid cross. Two traits, dihybrid cross. How do you make a Punnett square for that? How many potential gametes can my first parent make? The capital T, lowercase t, capital Y, lowercase y.
Can you repeat what they were called again?
Dihybrid cross. So let's think about gametes here. Say that these are on different chromosomes. So they're not located on the same chromosome, but they're on different chromosomes so they can assort independently. I could make one gamete from this first parent with a capital T, and that could pair with a capital Y, right? That's one potential combination of those letters. Or the capital T could have paired with the lowercase y. Agreed? Two potential gametes. But I still have to address what happens if it's a lowercase t and a capital Y. Or a lowercase t and a lowercase y. So how many possible gametes are there when we look at two traits? 4. How many possible gametes will the other parent make? 4. So instead of a Punnett square with 4 squares, 2 across the top because it makes 2 potential gametes, we make a Punnett square with 4 gametes across the top, the gametes from this parent, and 4 gametes down the side, the gametes of the other parent. So it would look something like this. Here's where I caution you when you set up a Punnett square. Make sure you always choose letters where the capital and lowercase are easily distinguished from each other. Really bad letters to use. O, C, X, W, U, S. Don't ever use those letters. You won't be able to tell the difference, even if you make one gigantic and one truly tiny. It gets very confusing very quickly. We agreed that was the first gamete, right?
Yep.
And then this was the second gamete. And then this was the third gamete. And the fourth. Other parent produces the same gametes. Challenge yourselves, friends, between you and your partner. See if you can't, one, fill out this chart, and then do a genotype and phenotype ratio for all of the potential offspring.